Okay, so this first webinar of the session is something about basic, which is what is science and what is science applied to the conservation of marine turtles. This first talk is something about science, and it is like an introduction of the other webinar, which will be more centered on marine turtles, the life history of marine turtles, and what we should do to better understand and to better conserve marine turtles. So for this first talk, this is the basis of what is the science of conservation. Okay, the science of conservation, conservation biology, is are two fields that are linked. The first field is about the true science, the conservation biology, which is the biology applied to conservation. Many different fields of biology are involved in conservation biology. It can be ecology at the level of ecosystem, ecology at the level of population, genetics, ecology of community, but also the social sciences, such as biogeography, sociology, environmental ethics. All this kind of science can be used to make conservation biology as a science. When you have defined something, when you have perhaps a solution, then you can apply what you have done with the field of science. Then this is the applied science. Then you must work with fishermen, fisheries management. You must work with authorities to manage the land use, for example. So the biology of conservation are really two parts, a scientific part and more applied science. Then, as we have just seen that conservation biology is a science, but also is an applied science, we should define clearly what is a science and how the concept of science can be applied to marine turtles. So now we will talk a little bit about what is a science and what is a scientific activity. Let's see first what is a science, then what is a scientific activity, and then how we can apply this to marine turtles. There are three ways that we can apply the knowledge to different situations. The three ways we can apply the knowledge are science, religion, and DOGSA. And we will see now what is precisely each of these concepts to know, to better define what is science and what is not science. Probably you know two of these names, science and religion. Perhaps DOGSA, you don't know exactly what it is, so we will precisely define this concept. DOGSA, this is both knowledge and misunderstanding. So the DOGSA is a set of false or confused belief founding the common truth. When you take a drink with friends and you talk about how the world is moving, how things are happening in the world with 3 or 4 beers. You use the DOXA. The DOXA, this is just a belief, but this is not based on the true knowledge. Example, we will take each time an example. An example of DOXA applied to marine turtles. When you say that the sea turtles were on the earth at the same time as the dinosaurs, but they survive. This is something that you can read in publication. But this is something that is so imprecise that you can do nothing with such a definition. Why this is imprecise and then it becomes wrong? First, in this statement, you make an equivalence between the marine turtles at current time and the origin of marine turtles. The marine turtles present on the Earth 60 million years ago are not the same as the marine turtles that you see now on your beaches. So this is 2 different concepts. And when you say that the sea turtles were on the Earth at the same time as the dinosaur, you forget that this is not at all the same marine turtles. Another problem with this sentence, you claim that the dinosaurs are no more present on the Earth, but birds are dinosaurs. Birds, from a systematic point of view, they are dinosaurs. So you cannot say that dinosaurs have disappeared because they are still present as a bird. So this sentence is so confused that it is not science. This is just DOXA. You can say this after 2 or 3 beers, but you cannot say this when you are doing science, because in science you must define clearly the concept you are using. Then the second term in the way we can use knowledge is religion. Religion is based on dogmas. A dogma is a point of doctrine that is regarded as a fundamental and unquestionable truth. And therefore, it cannot be challenged, it cannot be tested. The dogma is the origin, is a basis, not the origin, the dogma is the basis of religion. And then the dogma by nature is true. It cannot be tested. I show you here 2 dogmas. The first is a dogma, one of the dogma of Christian religion. The other is one dogma of the Muslim religion, but it cannot be tested. All the religion is built taking into account this is true. So, for example, if you say, with science, I will test if religion is true, it is a nonsense, because religion is based on dogma that cannot be tested. I will take an example on dogma applied to marine turtles. For example, when you read, sea turtles do not feed during the nesting season. You can read this, for example, in this very nice paper by Jeff Miller in 1997, Reproduction in Sea Turtles. And for many persons, this statement is seen as so obvious that it is no longer needed to be tested, it becomes a dogma. But when you look more precisely on the data, you see that this dogma that sea turtles do not feed during the nesting season, this dogma has never been really validated. So it becomes something that we say again and again and again, but it has not been tested. It becomes a dogma applied to marine turtles. And now we enter in the field that we will use for the next part of the talk, which is science. How to define science? This is very complicated. Science, the most accepted definition, has been proposed by Karl Popper, scientific epistemologue. Science should be an activity which should use only testable and refutable results. This is something very important, that when you are doing science, you must test, and following the test, you can prove it is wrong or not. This is main difference with two previous knowledge fields. This is a main difference because when you are doing science, all you say should be refutable. Refutable, it does not mean that it is wrong. It means that you can show that it is wrong. This is something very different as compared to dogma. The dogma, you cannot show it is wrong because all is built on the hypothesis that dogma is true. For science, there is no dogma. Each time you say something, you must be able to show that it is wrong. It can be true, but you must be able to show that it is wrong. Let's see how, from this definition of science, we go to the practical way we are doing science. Let's make a very short history of the history of science. The first person who can be named as a scientific is Aristotle. Aristotle is a philosopher of Greece, and Aristotle differs from his master, Plato. And the main difference between Plato and Aristotle is that Plato thinks, and from the thinking, he tried to get the truth. Aristotle said no. First, you must observe, and from the observation, you know the truth. The truth is not thinking, the truth is observation. This is a very big difference between Plato and Aristotle. Then another person very important in the concept of scientific activity is Alhazen. The true name is written at the top, but it is nearly impossible to say it. Alhazen was a scientific from Egypt or Iraq, from this Iran, Iraq, this region. And what Alhazen has done that was really new at that time was experiment. Each time Alhazen thought about something, he was doing experiment to test if his idea was correct. So we see that first Aristotle observed. Alassane makes experiment, and the link between both can be obtained with Roger Bacon. Roger Bacon was an English, and for Roger Bacon, you should always question what has been learned before. It was very important. At that time, there was some truth that you thought it was always true. Roger Bacon said, no, we should always question what we thought it was true. And it was the first who questioned what Aristotle has saying before. This has been synthesized by René Descartes in the Discourse of the Method. And René Descartes tried to explain the scientific method. The scientific method, this is a relationship between observations, theory, prediction, and experiment. And with this four way of thinking, you build the scientific method. When you are doing observation, you can try to generalize the observation, and you will make a theory. Using the theory, you can make predictions. Then from these predictions, you can make experiment to test the predictions. So you are doing experiment, different kind of experiment. We will see just after some different kind of experiment applied to marine turtles. And from the experiment, you are doing new observations. You will change a little bit or profoundly the theory, and so on. It makes something like a progress, a general progress of the way you are explaining the situation you are working. So the science is defined by the fact that each time you are saying something, it should be testable, and you should be able to show that it is wrong. This is the science. And the scientific method, this is these four ways to describe the world: observation, theory, prediction, experiment. Now we will see these four ways to make science and apply to marine turtles. First, when you are doing science applied to marine turtles, you can do some observations, some descriptions. Here you see that this is a beach. This is in French Guiana beach. This is some field. Okay. This is some field work that has been done. It was in French Guiana. It was for leatherback. For each leatherback, we record where was the change in the carapace or some illness, and we measure, we make a drawing of the head white color. The difficulty is if you stop here, this is not science. If you stop at the position where you describe the world, you cannot make generalization. There is a sentence that I like: the plural of anecdote is not data. This is anecdote. You take this drawing, this description for one individual, for another individual, for a third individual. I have in my lab approximately 1,000 such description. But if we stop here, we just describe the world, but this is not science, because to be science, from the observation, you must do theory, prediction, and experiment to test the theory and the experiment. With the experiment, you are doing new observation. So if you stop here, if just you describe the world around you, you are not doing science. For science, you must do also theory, prediction, experiment. What kind of experiment? I present here 3 different kinds of experiment: small, medium, or large-scale experiment, natural experiment, modeling. When we test hypotheses, so we make experiment at different scales. Here, this is a baby turtle, and we measure the rate at which the baby turtle is running. And it was to test if the condition of incubation of embryo make some change on their ability to escape more or less fast. This is an experiment, in this case, at small scale, but we can make experiment at different scales. Something that is perhaps less obvious for you, this is the natural experiment. What is a natural experiment? A natural experiment is an experiment where you do not manipulate by yourself the ecosystem. You observe the ecosystem in different situations. And from these situations, you can make as an experiment. The simplest way is that I show you something that we have done. It was in French Guiana. It was a student from the lab. And what we wanted to test is if the beach is stabilized by vegetation. And then we would like to know if the vegetation behind the beach permit to stabilize the beach and limit erosion. The hypothesis is that if we enrich the sand with nitrogen, vegetation will grow better and the beach will be stabilized. To do this is very simple. You put fertilizer at the back of the beach. With fertilizer, the vegetation will grow, and then you measure erosion in part of the beach with fertilizer and in part of the beach without fertilizer. This is the basic idea. The problem is that the beaches are protected and it is forbidden to manipulate the nitrogen contents of the beach. We didn't have the authorization to use fertilizer on the beach. I don't say it's bad, it was very good, but we cannot do the experiment to test if there is an impact of the vegetation. But when you look at this is a typical nesting season. This is a nesting season in another part of the world. This is in Guadeloupe, in Caribbean island. This is the nesting season of marine turtles, and you see that turtles bring eggs, in this case, from July to November. What what there is in X. In X, you have nitrogen, and when the turtles deposit eggs on the beach, they deposit nitrogen also with the egg. If the incubation is successful, the baby turtles leave the sand, goes to the sea, and then the nitrogen goes with the baby turtles to the sea. But if incubation is does not produce juveniles, if the egg die in the sand, then the nitrogen present in the egg stay on the beach. Then you can imagine this is the turtles that are doing the job for you. They bring nitrogen on the beach, and you can use the fact that turtles bring nitrogen to test the hypothesis that nitrogen change the vegetation and change the erosion. This is an natural experiment. You don't ask to turtles to come to deposit nitrogen. They are doing naturally, but what they are doing naturally can help you to solve a question about the impact of nitrogen on the vegetation and the impact of vegetation on the erosion. So this is a natural experiment. Here, when we come back here, the experiment, so we can do small, medium, large-scale experiment, natural experiment, but also modeling. Here, this is an example of modeling. This is a result that we have obtained with Jenny Tusek and Ronel Neil. They work in South Africa. And the main nesting beach is located at the border between South Africa and Mozambique. We were searching what was the, so we have 20 years of data. For 20 years, we know when the turtles begin to nest for each year. Sometimes they begin to nest a little bit early, sometimes they begin to nest a little bit late, and we search if in Indian Ocean we find a relationship between oceanographic conditions and the period of nesting here at the border between Mozambique and South Africa. And we find a very strong signal in this region. This is Mayotte Islands. We find a very strong signal. The red is a very strong signal from between oceanographic conditions and the nesting season here. Now we know that this is where the turtles are feeding. They feed here, and so when this is a very good season here, they come to nest earlier here. When this is a bad season here, they come to nest later. Then we have been able to visualize where the turtles are nesting, are feeding before to come to nest here at the border between South Africa and Mozambique. Ecology is a field of life science that use modeling the most. You cannot be an ecologist if you are not, at least at some level, a statistician or a modeler. You must have this culture. Why modeling is important in conservation science? First, it allows to manage great complexity of the studied system. Here we have turtle nesting here, and we have all the oceanography at the scale of Indian Ocean to take into account. Without modeling, it will be impossible to test some hypotheses at this scale. But also, the use of modeling allows you to make replicates, whereas you have only one Earth. You have only one Earth. There is no planet B. We have one Earth, but when you are doing science, you must do replicates, but you cannot do replicates because you have only one Indian Ocean. And we generate the replicates using modeling. To make a sort of synthesis of what is a science, what is not a science. So science versus pseudoscience. The science, when you are doing science, you make findings based on evidence collected. You make experiment, and from the experiment, you build theory and you build prediction. For pseudoscience, you start with a pre-established conclusion, and you try to confirm this pre-established conclusion. This is a very bad way to do science. Sometimes you can see this even in publication, but this is not a good way to do science. You must let the data talk. You must not try to make the data doing what you want. When you are doing science, you must take into account all the data to conclude. If you are doing pseudoscience, you select the data that support your belief. This is not, again, the good way to do. You must use clear, precise, technical vocabulary. If somebody tell you something very confused with sometimes scientific words, but you don't understand clearly the logic, this is pseudoscience. You must make a cautious statement, use conditional tense. For example, you will say, we could conclude that if you read something that with extraordinary claims without nuance or evidence, this is not science, this is pseudoscience. You must use a rigorous reproducible method that can be verified by the entire community. This is the principle of material and method in the publication. When you write a material and method, it must be precise, and each person should be able to do the same as you have done. With for pseudoscience, you use something that cannot be verified. When you say something, you must be sure that what you say is the best of what you understand at the point you are doing the job, but somebody will do better later. So you know that when you are doing science, what you are doing will be shown to be not the best we can do a little bit later. The pseudoscience is dogmatic and inflexible. You must accept criticism, and you accept to be refuted, because this is the basis of science. If you are doing pseudoscience, you do not accept to be criticism, and what you say cannot be refuted. So we work in the blue concept. We don't work with the red concept. Imagine that you want to apply this concept to the marine turtle conservation. For example, you try to respond to EV predation on sea turtle eggs. we see again this graph between observation, theory, prediction, and experiment. What you will do, you will evaluate the level of prediction on X. You will identify the causes, which can be human, physico-chemical factor, predators, or others. You will propose a method to reduce certain cause, and you will propose a method to evaluate the expected impact. You will implement the method. Then again, you evaluate the level of predation. You come back here. You come back here, you identify the causes, human, physico-chemical factors, predators, and others, and you test if the method that you have implemented gives the expected results. Is the predation lower as you have expected? Then you must use a test. A test is a method that is used in many fields in science or humanities. It consists of a comparison between an observation and a prediction. It is necessary to make sure that the observed value corresponds to a prediction of the model. And then it is necessary to know if the chance by itself could have produce something that you could, if it is not the chance, it is what you have done that has gave this result. Then we will talk just a little bit about statistical tests. I will not give formula, I will not explain you how to make tests because each test is different, but we'll talk a little bit about the logic of the test. When you are doing a test, generally you use what is called H0, H1 test. The H0 is called the null hypothesis. This is the hypothesis that is clearly formulated. For example, the H0 is there is no difference between predation before and prediction after in the previous example. So H0 is clearly defined. H1 is not clearly defined. H1 is all that is not H0. The name is alternative hypothesis. These are all the hypotheses that are not H0. Then, when you are doing the test, you will check if the data could have been obtained just by chance, or the probability that the chance gave such a level of difference is so low that you will conclude there is a true effect. And then, after doing the test, you generate a p-value. P-value, this is the probability of being wrong if you reject H0 as a valid hypothesis to explain your data. Let's take an example, it will be simpler. After doing the test, for example, you observed a p-value of 0.2. If you have such a value, it means that if you reject H0, remember H0 is clearly defined, so you can reject H0. If you reject H0, you know that this data set had 20% of chance of being obtained when H0 was true. So you are taking a large risk to reject H0 because the data have 20% of chance to have been obtained with H0. then in this case, you can say that you do not reject H0. Okay. This is, so the value, the p-value is what is called the first order risk. This is technically the probability of data knowing H0. Okay. But the p-value, the way we are doing tests, has some limits. Suppose that an article reports a p-value of 0.001. This p-level, this p-value, may be related to a not very strong effect in the population, but with a very large sample size, a large effect with a moderate sample size, or a very large effect with a small sample size. Then the p-value by itself does not tell you if the effect is strong or not. This is a combination between your sample size and the strength of the effect. Similarly, if you have a p-level of 0.075, it can be a very large effect, but with a small sample size, or a teeny effect with a very large sample size. So the p-value cannot be interpreted directly. You must always take care of what the p-value said in relationship with the sample size. So for this slide, we will read only this last sentence. The p-value gives us an indication if there is an effect or not, but this is not directly interpretable. So if the p-value is very small, probably you have a significant effect, but you must take care that the sample size can make something more confused. So the p-value gives an indication, but it is not a dogma. This is an indication. Let's take a real example. This is a work done by Adriana Cortez. Adriana Cortez is an ecotoxicologue. She works about marine turtles in Mexico. And so she works on olive ridley turtles, and she has found something very strange. Let me explain you. She studied biological marker. And so the biological marker, this is what is called biomarker. And she studies the relationship between this marker and the contamination of individuals. If individuals are contaminated, do they present some gene expression? She studied the SOD, which is a superoxide dismutase. This is an enzyme that reduces the impact of contamination. She studied the gene expression of SOD and the activity of the protein. Normally, for those who have done biology, you remember you have DNA, transcription, the mRNA goes out of the cell, then it is translated into protein. The protein is a SOD, and the gene expression, this is the mRNA of SOD. And there was no relationship between the quantity of mRNA and the activity of the protein. You see here, we see P 0.2, 0.1, 0.5, let's say. So she does not find a relationship between quantity of RNA, quantity of protein. This is strange. Normally, we should see a relationship. So if she does not see something, perhaps it is because really there is no relationship, because it can be the regulation can be very complicated, or perhaps because the number of embryos tested were too low. So it was not embryo, it was adult, sorry. So you see that p-value give you some information, but are very complicated to clearly interpret. To come back to the conservation applied to marine turtles, conservation biology must behave like any science, and any action must be evaluated. You cannot do conservation biology without the scientific method, without theory, prediction, and test of the prediction using experiment or new observations. So conservation biology applied to marine turtles is a science and must behave like a science. Okay, that's all for today. It is 2:30.